वेलकम बैक टू द चैनल। वी आर स्टार्टिंग अवर न्यू सीरीज ऑफ ग्लूकोज मेटाबॉलिज्म। क्या आपने ये सोचा है कि जितना भी फूड हम इंटेक करते हैं उससे एनर्जी या एटीपी कैसे बनती है? उसके लिए हमें सबसे पहले क्रेब साइकिल के बारे में जानना पड़ेगा। जो कुछ भी हमने खाया वो एक कॉमन कंपाउंड एसिटाइल कोए के थ्रू लिंक हो रहा है क्रेब साइकिल में। इन सिंपल वर्ड वी कैन से दैट क्रेब साइकिल इज द सेंटर ऑफ ऑल मेटाबॉलिज्म बाय विच वी जनरेट्स एनर्जी। सो लेट गेट स्टार्टेड। पहले हम ओवरव्यू ले लेते हैं साइकिल का। क्रेब साइकिल का ये है कि सबसे फर्स्ट एसिटाइल को है जो कि 2 कार्बन कंपाउंड है, वो कंडेंस होगा 4 कार्बन कंपाउंड ऑक्सैलो एसिटेट के साथ टू फॉर्म अ सिट्रेट। देन दिस सिट्रेट विल चेंज टू आइसोसिट्रेट। आइसोसिट्रेट फर्दर react to form alpha ketoglutarate. Alpha ketoglutarate then change to succinyl CoA. Succinyl CoA then convert into succinate, then fumarate, then malate. Malate will regenerate oxaloacetate. This is the overview of a cycle. Now we move to the reactions of the cycle. Oxaloacetate is first condensed with acetyl CoA and then is regenerated as the cycle is completed. 2 carbon enter the cycle. 2 carbon is our acetyl CoA which enters the cycle as acetyl CoA and moves out the cycle as 2 CO2s. So we can say that in this cycle there is no net production or consumption of intermediates. Okay, now we move to acetyl CoA production. The major source of acetyl CoA are glucose, pyruvate, fatty acid and ketogenic amino acid. And the fate of acetyl CoA is oxidation to TCA cycle or fatty acid de novo synthesis or cholesterol synthesis and ketone body synthesis. In fatty acid de novo synthesis, the first molecule comes as acetyl CoA, then further molecule enter the process as a malonyl CoA, which we further study in our further more videos. Now, major source of acetyl CoA for TCA cycle. The major source for acetyl CoA for TCA cycle is our pyruvate, which comes there are many sources from which we obtain pyruvate. For example, glycolysis. In glycolysis, pyruvate form 2 moles of pyruvates are formed. In this pyruvate, on this pyruvate, pyruvate dehydrogenase complex, which is a multi-enzyme complex, acts and form acetyl CoA. And this reaction is also known as link reaction, which links our glycolysis and Krebs cycle. As we know that pyruvate dehydrogenase complex, it is not a component of TCA cycle, and this reaction takes place in mitochondrial matrix. Now we study about the PDHC, which stands for pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex is a protein aggregate of multiple copies of 3 enzymes, which include E1, pyruvate decarboxylase, E2, dihydrolipoyl triacetylase, E3, dihydrolipoyl dehydrogenase. Each catalyzes a part of reaction. Their physical association links the reaction in proper sequence without release of intermediates. तो समझिए ऐसा है कि अगर E1 ने रिएक्शन को कैटेलाइज कर दिया तो प्रोडक्ट बनकर ही छूटेगा जब तक E3 उसको कैटेलाइज ना कर दें। उनका एसोसिएशन ही ऐसा अरेंज है, लाइक दिस E1, E2 एंड E3 substrates come, E1 react on it, then E2, then E3, and there is release of product. And this enzyme is regulated by 2 enzymes known as pyruvate dehydrogenase kinase, pyruvate dehydrogenase phosphatase. Now, coenzymes of pyruvate dehydrogenase complex. 5 coenzymes act as oxidant or carrier for the intermediates. Now let us know about the coenzymes. These coenzymes are, as we know that the vitamins act as a coenzyme. Then coenzyme for E1 is thymine pyrophosphate, which is an B1 complex. Second, E2. For E2, coenzymes are lipoic acid and coenzyme A. For E3, coenzymes are NAD, FAD. NAD stands for niacin adenine dinucleotide, flavin adenine dinucleotide. This comes from niacin and this from riboflavin. These both are vitamins. Now, TPP, lipoic acid and FAD tightly bound to the enzyme and function as a prosthetic group. This is an important point that we have to remember, that TPP, lipoic acid and FAD are tightly bound to the enzyme and act as a prosthetic group. Mechanism of action of pyruvate dehydrogenase complex. First of all, pyruvate enters, on which TPP acts, and TPP acts. The enzyme is pyruvate decarboxylase, which is E1. There is decarboxylation, removal of CO2, and linking of TPP to pyruvate. Pyruvate is decarboxylated to form a hydroxyethyl derivative bound to reactive carbon of TPP. TPP is bound to this remnant of pyruvate through reactive carbon. Now, second reaction. The hydroethyl intermediate is oxidized. This is the hydroethyl intermediate is oxidized by enzyme dihydrolipol transacetylase, which is also known as E2. The hydro is oxidized by transfer of disulfide form of lipoic acid covalently bonded to E2. This is disulfide form of lipoic acid. It's transferred to hydroxyethyl intermediate to form Now, second reaction. The hydroethyl intermediate is oxidized. This is our hydroethyl intermediate, which is oxidized by transfer of disulfide form of lipoic acid covalently bonded to E2. This is our disulfide form of lipoic acid, which is covalently bounded to our E2. Then third reaction, the acetyl group bounded as a thioester to the side chain of lipoic acid is transferred to CoA. This is our acetyl group bounded as a thioester to the side chain of lipoic acid, which is transferred to CoA for the formation of our acetyl CoA. Now, we have to regenerate our disulfide form of lipoic acid, for which the sulfhydryl form of lipoic acid is oxidized by FAD dependent E3 to the disulfide form. For this, dehydrogenase act on sulfhydryl form of lipoic acid to convert into disulfide form of lipoic acid. In this reaction, FADH2 is formed. The FADH2 is further oxidized by oxidized NAD plus to FAD, and it itself converting into NADH. Now, the regulation of PDHC. Pyruvate dehydrogenase complex, as we discussed, that it is regulated by two enzymes: pyruvate dehydrogenase kinase, pyruvate dehydrogenase phosphatase. Pyruvate dehydrogenase kinase, what it does? It does phosphorylation of this multi-enzyme complex. And PDH phosphate, what it does? It does dephosphorylation of our multi-enzyme complex. Then this is a basic concept. Phosphorylation makes any enzyme inactive, and dephosphorylation makes it active. So the kinase activity makes our multi-enzyme complex inactive, and phosphatase activity makes our multi-enzyme complex active. Now, the PDH kinase is positively regulated by product of the reaction like ATP, acetyl CoA and NADH, and it is inhibited by pyruvate. PDH phosphatase is positively regulated by calcium plus 2. Now, this is our enzyme regulation. This enzyme is further regulated by product inhibition. The product of the reaction, acetyl CoA and NADH, negatively regulate our active enzyme. Calcium plus 2 is a structure strong activator of PDH phosphatase, stimulating even activity. This is particularly important in skeletal muscle, where calcium plus 2 release during contraction stimulate pyruvate dehydrogenase complex, thus energy production. As we know that during our muscle contraction, calcium is released. For muscle contraction, we also need required ATP. So this that calcium released is also acts on our PDH phosphatase so that our enzyme can get active. The first deficiency we discuss about this is congenital lactic acidosis. E1 of PDHC is tetrameric, in which deficiency of alpha 1 subunit is most common cause of lactic acidosis. E1 is formed by 4 tetramers or tetramers in which there is a deficiency of alpha subunit, which causes its inactivity. So that's why pyruvate isn't get converted to acetyl CoA. Pyruvate accumulation leads to anaerobic oxidation of pyruvate to lactate by enzyme known as lactate dehydrogenase. This creates particular problem for brain because for brain it mainly relies upon TCA for energy, and acidosis it is more sensitive. Our brain is more sensitive towards acidosis, and this disease is our X-linked dominant disease. Next, arsenic poisoning. Pentavalent arsenate can interfere glycolysis at glucose 6-phosphate step. We will discuss about this in our further videos. Arsenic poisoning is due to inhibition of enzyme complex that requires lipoic acid. Now, lipoic acid is required by pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase complex, and branch chain amino acid dehydrogenase. In all three reactions, lipoic acid is required as an coenzyme. So, arsenic can cause poisoning in these steps. It forms a stable product with thiol group of lipoic acid, making compound unavailable to work as a coenzyme. Now we discuss our reactions of our TCA cycle. First reaction, formation of citrate. Oxaloacetate, which is 4 carbon compound, condenses with 2 carbon compound acetyl CoA to form citrate. The enzyme used is citrate synthase. The standard change in free energy of this reaction is highly negative, which strongly favors the formation of citrate, and it is a type of aldol condensation. This enzyme is inhibited by citrate, means it is an example of product inhibition. When oxaloacetate binds to citrate synthase, its affinity towards acetyl CoA increases. This is a type of regulation. What does it says? That oxaloacetate when binds to our enzyme citrate synthase, its affinity towards acetyl CoA increases. Now citrate. About citrate, we have to know about this citrate, that it is an intermediate of TCA cycle. Yes. Source of acetyl CoA in cytosol for fatty acid and cholesterol synthesis. And it inhibits phosphofructokinase, rate limiting step of glycolysis. It activates acetyl CoA carboxylase, rate limiting step of fatty acid synthesis. Now it's all about citrate. Now we further moves towards our citrate isomerization. Citrate is isomerized to isocitrate. In this, there is a migration of hydroxyl group from migration of hydroxyl group. Fluoroacetate is a potent inhibitor of aconitase. The enzyme required for this conversion is aconitase, which is also known as aconitase hydratase, which is an iron sulfur protein. The fluoroacetate is converted to fluoroacetyl CoA, which contains with oxaloacetate to form fluorocitrate. This fluorocitrate is a potent inhibitor of aconitase. So that's why we when we collect our blood, we uses our fluoride vials. Next reaction, oxidative decarboxylation of isocitrate to form alpha ketoglutarate. Isocitrate is oxidatively decarboxylated to alpha ketoglutarate, in which there is a oxidation and decarboxylation. The enzyme required is isocitrate dehydrogenase. This is our rate limiting step of TC cycle, and this reaction is positively regulated by ADP and calcium and negatively regulated by ATP and NADH. Next reaction, oxidative decarboxylation of alpha ketoglutarate. Remember, in these two reactions there is decarboxylation. Only the two reactions in which there is decarboxylation. Of which compound? That is our acetyl CoA, which enters the 2-carbon compound and leaves the cycle as a 2-carbon, one CO2 here and one CO2 here. Alpha ketoglutarate is converted to succinyl CoA, and enzyme required alpha ketoglutarate dehydrogenase, in which there is oxidation and decarboxylation. This is negatively regulated by NADH and succinyl CoA, product inhibition, and positively regulated by calcium. Alpha ketodehydrogenase, it is a multi-enzyme complex in which 3 enzymes, and a mechanism of action is same as a pyruvate dehydrogenase complex. This enzyme requires 5 coenzymes as I discussed earlier, and the large change in free energy favors the formation of succinyl CoA. However, it is not regulated by phosphorylation and dephosphorylation. As we discussed that phosphorylation of pyruvate dehydrogenase complex inactivates the enzyme and dephosphorylation activates the enzyme. There is no such concept in this enzyme complex. Alpha ketoglutarate is also produced by oxidative deamination and transamination of amino acid. In urea cycle we have discussed this point, so check that video for this. Now next reaction, succinyl CoA cleavage. Succinyl CoA is cleaved to succinate, which is a reversible reaction. The enzyme required is succinyl CoA synthetase. At this step, substrate level phosphorylation occurs and there is a formation of GTP, which is coupled to form ATP. Source of succinyl CoA. First, propionyl CoA by oxidation of OH chain amino acid. When OH chain amino acid are beta oxidated, there is formation of, at last step, there is a formation of acetyl CoA plus propionyl CoA. This propionyl CoA is converted to succinyl CoA. Metabolism of several amino acid. Several amino acid, specifically which amino acid? Glucogenic amino acid. Then it can be converted to pyruvate for gluconeogenesis and used in heme synthesis. Okay. Next step, succinate oxidation. Succinate is oxidized to fumarate with the help of enzyme succinate dehydrogenase. At this step, FADH2 is formed, and our reducing equivalent is FAD, flavin adenine dinucleotide. Succinate dehydrogenase is embedded in intermitochondrial membrane. It functions as complex 2 of ETC. For this reaction, you have to check our ETC. In that reaction, our complex 2, succinate dehydrogenase, that is this enzyme. Succinate dehydrogenase is not able to reduce NAD+, so that's why it uses FAD as a reducing equivalent. Next step, fumarate dehydration. Fumarate is dehydrated to malate by enzyme known as fumarase. Source of this fumarate is first urea cycle, second purine synthesis and catabolism of phenylalanine and tyrosine. Next reaction, malate oxidation. Malate is oxidized with the help of malate dehydrogenase to oxaloacetate. In this reaction, our oxaloacetic acid is regenerated. But change in free energy is positive for this reaction. Instead of this reaction moves in forward direction. Why? Because the reaction is driven in forward direction because of highly exergonic reaction of citrate synthase. This oxaloacetate will combine with acetyl CoA to form citrate, and this reaction is driven by citrate synthase. This citrate synthesis has highly exergonic effect, so that's why this reaction moves in forward direction. Oxalic acetate is also produced by transamination of aspartate. Okay. Now, energy calculation of TC cycle. 4 pairs of electrons are transferred during 1 turn of cycle. 3 4 NAD plus and 1 4 FAD. Now let us check where NAD plus is formed and where FAD is formed. In this reaction, 1 mole of NADH is formed. At this step, 1 mole of NADH is formed. At this step, 1 mole of FADH2 is formed. At this step, 1 mole of NADH is formed. Now, 1, 2, 3. 3 NADH is formed and 1 FADH2 is formed here, and substrate level phosphorylation occurs in this step and forms GTP, which is coupled to ATP. So total 4 pairs of electrons are transferred to NAD plus and FAD. In this reaction, 3 NADH is formed, which is equivalent to 7.5 ATP. 1 FADH2 is formed, which is equivalent to 1.5 ATP, and 1 substrate level phosphorylation, that GTP is formed, is coupled to ATP. So there is a net production of 10 ATP per cycle. Thank you.
Audio Jungle.